So, have you ever stopped to think about your hands? Why your left hand fits a left glove, but, not a right one?
Sure, yeah. It seems obvious, but there's deep chemistry there.
Exactly. And what if life itself had that kind of fundamental handedness? That's what we're diving into today, mirror life.
Right, hypothetical life built from molecules that are perfect mirror images of the ones we find here on Earth.
Our mission is to dig into the science, the reports, the policy debates, basically understand the foundations, the potential, and also the pretty serious risks.
It's a fascinating area.
So the big question is, what happens when we can actually build life as its perfect reflection? Why is that both so promising and, frankly, kind of terrifying?
To really get it, we need to start with chirality. That hand analogy is perfect. They're mirror images, right? But you can't lay one perfectly flat on top of the other.
Non-superimposable.
Precisely. In chemistry, those mirror image molecules are called enantiomers. And life on Earth, well, it shows what we call homochirality, meaning it almost universally uses one hand. Amino acids for proteins, D sugars for DNA and RNA. It's like a fundamental choice life made.
And how critical is that choice? Is it just a quirk or is it essential?
Oh, it's absolutely critical. Think of enzymes as locks. Only the correctly handed key, the right molecule shape, fits.
Okay.
Take carvone. The L form smells like mint, but its mirror image, the D form, smells like caraway. Totally different interaction with our receptors.
Wow, just from being a mirror image.
And sometimes the consequences are much more severe. Remember thalidomide? One enantiomer was a sedative. Its mirror image caused terrible birth defects.
Right, a tragic example.
So this homochirality, this preference, it's not just functional, it's a potential biosignature. If we found that on Mars, say, huge news.
Which makes it kind of ironic that we're now thinking about building it here on Earth.
Exactly. It's a real paradox.
And this whole idea, it's not brand new, is it?
No, not at all. It actually goes back to Louis Pasteur in the 1840s. He discovered molecular chirality and even speculated, I think around 1860, about a whole mirror world of biology.
And then Sci-Fi picked it up.
Yeah, Arthur C. Clarke's story, Technical Error in 1950, brought it to a wider audience. But the science took longer.
When was the big breakthrough then?
I'd say 1992 at Scripps Research. They chemically synthesized a mirror image version of the HIV protease, a D protein.
Okay, and what did it do?
Well, it worked perfectly, but only on mirror image D peptides. It completely ignored the natural L peptides. They called it reciprocal chiral specificity.
So like a reverse key for a reverse lock.
Exactly. That was the proof of principle. Mirror proteins could function, but orthogonally, separately from natural biology.
And things have really sped up since then.
Massively. Dr. Ting Zhu's lab, among others, has been just systematically rebuilding life's central processes in mirror form. LDNA, lRNA, D proteins.
They made mirror enzymes.
Yes, mirror DNA polymerases between 2016 and 2021. Then a mirror Key7 RNA polymerase in 2022, step by step.
And that experiment with Pasteur's quote.
Oh, that was brilliant. They encoded Pasteur's quote, "Il est possible que l'univers soit dissymétrique," into LDNA, basically mirror DNA. Put it in pond water. A year later, it was still perfectly readable. The natural ddNA they put alongside it, gone in less than a day.
Incredible stability. You can see the potential for data storage right there.
Absolutely, bioorthogonal information storage, but there's still a big hurdle.
Which is?
The mirror ribosome paradox. Ribosomes make proteins, right? Well, to make lots of mirror D proteins efficiently, you need a mirror ribosome, which itself is made of mirror RNA and mirror proteins.
Ah, chicken and egg problem. You need the mirror machine to make the mirror parts for the machine.
Precisely. That's the major bottleneck for building a fully autonomous mirror cell.
Is this where something like AI comes in? Can it help crack this?
It's certainly accelerating things. AI, particularly these new generative models, they're revolutionizing how we design proteins. Well, they can learn the fundamental rules of protein folding, which are independent of chirality, of handedness. So AI can design D proteins, mirror proteins, even without loads of D protein training data.
So it can design for stability, function, even synthesizability, helping overcome that synthesis bottleneck you mentioned.
Exactly. It speeds up the whole design, build, test cycle enormously. It's a game changer.
Okay, so let's talk applications, the non-replicating stuff first. What's the promise there?
Huge promise in therapeutics. Because mirror molecules like D-peptides or nucleic acids are invisible to our natural L-protein-based enzymes.
They don't get broken down.
Right. So they last much longer in the body. Potentially less immunogenic, too. Think mirror image drugs, Spiegel mirrors, which are lRNA aptamers for targeting cancer, or D-peptides as antibiotics that bacteria haven't evolved resistance to.
Longer lasting, maybe safer.
Yeah.
Sounds great. And the data storage aspect.
Yes, that's super stable LDNA. But again, this is all for components, things that don't copy themselves.
Because the moment you get a self-replicating mirror organism, things change dramatically.
Right. That's the peril. It's potentially an existential risk. Think about it. A stealth pathogen.
Stealth, because?
Because our immune system evolved over billions of years to recognize L-amino acids and D-sugars, would likely just not see it. Neither innate nor adaptive immunity might work.
So it could replicate completely unchecked inside us, untreatable.
That's the fear. And if it got out into the environment...
The ultimate invasive species.
Pretty much. Nothing eats it, no natural phages infect it, antibiotics probably wouldn't touch it. It could outcompete natural microbes, disrupt food webs, nutrient cycles. Imagine a mirror photosynthetic cyanobacterium changing the atmosphere. It's hard to even model.
And once it's out, you can't recall it.
It's the definition of a Pandora's box. Irreversible. It would evolve, adapt, spread.
Which is why we saw that big call from scientists recently.
Yes, the Science article in December 2024, around 40 leading researchers, Nobel laureates like Jack Szostak, pioneers in the field like George Church, Kate Adamala calling for a global moratorium on creating self-replicating mirror life.
They compared it to Asilomar, but noted it's different this time.
Right. Asilomar was reacting to recombinant DNA technology already existing. This is anticipatory, trying to put guardrails up before we cross a potentially irreversible threshold.
Is there a deeper philosophical point they're making too?
There is. They talk about the coherence boundary, the idea that Earth's biosphere has this deep, evolved chiral unity. Introducing a self-replicating counter-chiral system is an ontological intrusion. It fundamentally breaks that coherence in a way we can't undo.
So it's this incredible duality. Non-replicating mirror parts could revolutionize medicine and data storage.
Huge benefits, potentially.
But self-replicating mirror life poses these profound, maybe planetary-scale, irreversible risks.
Exactly. That's the tightrope we're walking.
And this call for a moratorium, it's the scientific community trying to regulate itself proactively.
It's a crucial moment for responsible innovation.
It really forces you, the listener, to think, doesn't it? The Mirror World Challenge isn't just about what we can build technologically. It forces us to ask what we should build. How do we navigate this ultimate test of foresight, maybe humility, for the only biosphere we have?